A very simple numerical, let's understand. If map distance between genes A and B is 3 units and between genes B and C is 10 units, between C and A is 7 units, then find their location on the chromosome. Please remember one thing, gene mapping, this method of gene mapping is only possible on a linear chromosome. So when it comes to circular chromosome, or you can say the nucleoid of prokaryotes, it's not possible because it's a circular thing. But for a linear arrangement, yes, it is very easy to do these calculation. So the question says the distance between A and B is 3, B and C is 10, C and A is 7. So if you try to put them in a sequence like this, A, B, C, then 3 and 10, and this will be 13. But the question says the distance between A and C is 7. So this type of arrangement in sequence is not possible. That means let's try the other way out. A and B has a distance of 3, A and C has 7, we know, so distance between B and C will be 10. So this is the way the genes are arranged on the linear chromosome. So this is the easiest numerical which I have solved. I will be trying to do more slightly tough one in the next classes. You also try to practice these things. They are all available online also, or in your help books if you have at home. So the distance can easily be measured, and this distance help us to arrange the genes on a chromosome. So what is a chromosome map or a gene mapping? What is gene mapping on a chromosome map? A linear graphic representation. Remember, linear. So you cannot do this thing with a circular type of chromosome or genetic material. A linear graphic representation of the sequence and the relative distance between various genes on a chromosome is called as chromosome map, or is called as, this technique is called as gene mapping. And then Alfred Sturtevant in 1911 and 13 gave the complete gene mapping or the chromosome maps of Drosophila. So he took time. He once performed in 11 for some of the chromosomes, because there are 4 chromosomes, if you remember, 4 pairs. So 4 chromosomes are there. So for 2 chromosomes he did in 11, and by 13 he finished all. So now what is the importance of this? On chromosome, the genes are in linear manner, please remember. So they are the linear arrangement representation only. Then location of the genes on the chromosomes are very clear with the help of this concept. The sequence of the gene we learned today in this numerical, A, B and C. If you put like this, the sequence is wrong. The sequence is but C, A and B is the correct sequence. So this distance help us to understand the sequence also. So this whole represents the chromosome which carries the genes in linear manner. Then they are located in a particular sequence. And then the strength of linkage also, because when 2 genes are far, like these 2 are farther and these 2 are closer. So when 2 genes are close, chances of crossing over is less, or linkage is high. And when 2 genes are far, chances of crossing over is high comparatively, and the linkage degree is less. So this is all because of the linkage we are able to understand. Now let's come to the next topic, and that is the Morgan's experiment. So this will be slightly confusing because now we are going to use different type of symbols. And remember, we have 2 types of chromosomes here, XX chromosome in female and XY chromosome in the case of male. This is explained in the NCERT also, but every year I found students are confused with the NCERT explanation. So I have tried my own level to represent in an easier manner. First I will try to show you the experiment from the NCERT. This page of the NCERT I am referring to, page number 84. So these are the 2 crosses which have been explained in the NCERT, and these crosses are regarding Drosophila, 2 characters each time, done by Morgan, and how these proportions, instead of 1:1:1 ratio, they came out to be different. So let's understand from the notes which I have made. So what was the Morgan's experiment? Morgan took a female. Now remember, every time there is reproduction going on, we all know mutants are possible. Like out of thousands it is possible one could be mutant, or out of 10,000 maybe one could be mutant. So when Morgan was performing experiment with the Drosophila intensively, sometimes he found there are mutant individuals also, mutant male also possible, mutant female also possible. So what he did, he performed an experiment with a wild. Now when I say wild, remember wild means dominant, or wild means which is commonly found in the population. So there are two types of term we are going to use now, wild and mutant. And when you put a plus, plus represents the wild type and without plus represents the mutant type, because Morgan used different types of symbols. Now one more thing. In the case of Mendel, we used capital T for tall and small t for the dwarf. In this case, it is opposite. What Morgan did was, mutant is yellow. So Y, that's why Y is used everywhere for the color of the body. And when you put a Y plus there, so the Y plus means wild type and it is brown in color. So you are not using capital B and small b. Capital B according to Mendel should have been brown color and small b should have been the yellow color. But in this case he used the symbols of the recessive type or mutant type. So the symbol will be yellow body. This will be yellow body plus 1. So when you say plus, plus means wild type is which one? Brown body. So Y plus means brown body, Y means yellow body. In the same manner, W plus means red eyes, because it is a wild one again, red eyes, and W means white eye. So he used the alphabets of recessive form. Remember, Mendel used the alphabets of the tall form. So this is the difference. And plus means wild type, without plus means mutant type. So he performed crosses, and the cross was between male and female, and the male was wild type and the female was recessive type.